And next we have Sudha Seshadri.
Good evening, everybody. So I'm just so honored to be here today. I had to be here. Bruce has been a guiding light for my career. Also, a little intimidated after all these speakers and intimidated by a little virus, which is why I've been a little antisocial, hopefully. It has been, the cardiovascular health study, as you've all heard today, has been a pioneer. And one of the things it clearly emphasized was that it takes a village to develop, grow, and bring to life answers that are true and that stand the test of time. So when I joined Framingham, we thought it was a very large cohort, 6 cohorts, 15,000 people. And then 2007, as I was beginning an independent academic career, we got GWAS, and suddenly it was a tiny sample. So April 2007, I was given the job as one of the junior people to talk to the cardiovascular health study that was getting their GWAS, and to Rotterdam that was getting their GWAS, and trying to schedule my calendar so that 1 o'clock was the CHS, and then 8 o'clock maybe was the Rotterdam. And so one of these calls, Bruce is like, why are we doing this? Why don't you just put everybody together on one call? And of course, the first meeting, people are, I'll bring my data on a hard disk, you bring yours on a hard disk, and we look at it at the same time. But over time, of course, I'm going to just touch upon how the CHS principles served to imbue the creation of charge. They are the inspiration and soul of charge. And then in our own area, how over the last 15 years, charge has contributed in key ways to our understanding of the biology of brain health. And then how the charge principles within the neurology working group that I'm a part of have led so far beyond GWAS to being the principles of collaborative research in neurology of aging. So, of course, and the baby tiger of neurocharge was born. In the middle, you have the group that got together in 2008 in Boston University. And as many of you have heard again and again, at that time, Alzheimer research was not the way the cohort research was. It was very top down. And one of the things we brought to it was the charge principles. We had Josh, who was even younger than me, but just as confident as he is today. And it was very clear that this was not going to be top down, at least for charge. Charge was a coalition of the willing. These were not cats herded together with a common food bowl. These were cats that actually enjoyed gamboling together in the snow. It was based on fair principles of trust and transparency, but it was also flexible and pragmatic, right? You've heard about how we had to do meta-analysis, but Ken Rice and Tom Lumley also showed us that actually meta-analysis is better sometimes. When you have one study that began 1990 and is looking at white matter hyperintensity using the CHS rating scale, and another one that began 2010 and is using a different quantitative method, so long as every cohort was ranking people the same way, within that cohort, meta-analysis would work, whereas pooling it might not. And that people shared and cared. Suddenly, if you were an investigator in Framingham, like my brilliant colleague Stephanie DeBette was, she didn't have just one middling mentor. She had 50 mentors from all over the world. And her very first paper on subclinical infarcts, she had a mentor across the country in Will Longstreth at Charge and CHS. And that was one of the first neuro papers that Charge came up together. And of course, the principle of championing the young champions. When you started on something, it was like, who's going to be the young champion for this? And that has continued to lead to productivity. And we have learned to be stronger and smarter together, right? The first paper, we each looked at it in different cohorts, and each was getting a different answer. And then when you got a meta-analysis, you found that the answer was much more reliable and reproducible. So we broke away from the initial model of having half the cohorts being for discovery and half being for replication, and said, the strongest answer is the one that is the most together. So what have been some charge contributions? Before the first charge paper, the studies were mostly in one cohort. And the platform, it was maybe Affymetrix in one study, Illumina in another study. So using HapMap, we got together with an imputed GWAS. And again, because cohorts, you heard all this discussion about, should we take prevalent disease? Should we not take prevalent disease? But clearly one of the strengths of cohorts was incident disease. And for something like stroke, where 30% of people died in the short period after they got a stroke, we thought it was important to look at incident disease, which would show people with severe disease as well. And following on that study, over time, we inched up. We had about 12 loci, and around that time, FOXF2 was shown to be the first locus that clearly linked this VCID that all of us knew was important for dementia to the Alzheimer's that the rest of the world said was important for dementia. There were people in charge who would say, without vascular disease, amyloid is not leading to dementia. And there were people elsewhere who would say, without amyloid, vascular disease is not leading to dementia. But here, there was evidence that with the same amount of amyloid, if you had breakdown in the blood-brain barrier, you were more likely to have MCI. And here was a gene called FOXF2 that was expressed in the pericyte, clearly important to the blood-brain barrier. You see if the FOXF2 knockout, you see those hemorrhages in the little zebrafish, you see that methylene blue leaking out. And so genetics was showing us something we had never suspected before. We were trying to put together, as Jerry spoke about, polygenic risk scores. We had 12 genes for stroke. So we put together another 342 loci for different things that we knew increased the risk of stroke. And this was Carla. I think she may be one of the only people who has a tiger twice. And but at that time, we were not getting much addition to the clinical risk score. But over time, as it grew to be a megastroke, and you began seeing that there were types, loci associated with certain types of stroke, loci associated with certain risk factors, you could then create the pathway-specific risk scores that Jerry was talking about. And more recently, as we combined with the VA and moved from an earlier era, this was still only non-Hispanic whites because we were just beginning to learn how to analyze data with multi-ancestry. We were finding that you had the purple is the European ancestry, the green is the African American, and the brown is the Asian ancestry. And in every ancestry now, these polygenic risk scores were clearly predicting what the future was going to be. They could be used as a tool for gene-environment interaction, for targeted preventive interventions well before the age at which we would expect, say, brain health to deteriorate, and potentially in differential diagnosis as well. What about dementia? It was around 2009 that the first GWAS loci were identified. Charge was replicating some of those, but those were all non-imputed. And so some loci, like this locus called BIN1 and others, wouldn't show up if you pooled. So charge was the first one where it was based on meta-analysis, imputation, and showed this locus BIN1, which is the second biggest locus for late onset Alzheimer's after ApoE. And one of the early studies showed that BIN1 was actually increasing in expression in the oligodendroglia. That was not considered very important in Alzheimer's at that time. But today, with these amyloid-lowering medications, the importance of ARIA, we have recognized that oligodendroglia also have an important role in Alzheimer's. And over time, charge became the team with which all the cohorts went into every other collaboration. And so one of these collaborations was the International Genomics of Alzheimer project. Back in 2010, it began. And then this paper showed about 30 loci, including now the emergence. Till then, we were thinking mostly that we'd expect these genes to be expressed in neurons or their supporting cells, the astroglia. And so one of the interesting things within CHARGE, again, using incident AD, was in the Icelandic cohort, seeing this new gene called TM2D3, and a young investigator who was working with flies and showed that if you knocked out the fly analog, the flies wouldn't form. There was embryonic death. But if you put the human variant in there, the good human variant, the flies survived. With the bad human variant, they did not. And Josh is now a key part of CHARGE. This was his first major foray into that area, but now he is doing fly-based, studying so many potential variants. So in parallel with the GWAS, one of the things that became clear is that some of the GWAS hits were not expressed in either the neuron or the astrocytes, but were expressed in the microglia, which then completely changed the emphasis in Alzheimer's into the clearance of the amyloid, whereas in the APP understanding of early onset Alzheimer's, it was all about accumulation of the amyloid. And so we started looking for rare variants. Josh was leading this paper. Within CHARGE, CHS being the oldest cohort, had the largest number of people with Alzheimer's, and then Rotterdam, and then replicating in other cohorts as well. We were able to, again, substantiate the role of the immune response, the role of the microglia, and the clearance of the amyloid, and we're beginning to develop polygenic risk scores, which could help with disease and pathway-specific PRS. And one of the important things, which is still important today, we had a Nature Medicine paper saying E44 is like an autosomal dominant variant because everybody is going to get the disease. That's not true. Here, this was data from Rotterdam that showed that if you were E44, your risk could vary from 5% at a young age to much higher. But if you had one copy of E4, again, your risk could be throughout till 85, just as low as 5% or as high as 40%. And over time, you had this Belinguis paper cited as one of the top CHS cited papers. Over time, polygenic risk scores started predicting change from normal to Alzheimer's, as well as change from MCI to Alzheimer's. And more recently, looking at this combined entity, the commonest cause of dementia is all-cause dementia in cohorts. And so MEGA-VCID, a sample of nearly 1 million people, small compared to the diabetes or hyperlipidemia studies, but quite large, is beginning to show what may be the genetic variation, predicting risk as well as resilience to dementia. But regardless of the genes, environmental factors matter. I'm not talking about today about social contextual factors. And with the same genetic risk, you could halve your risk. One of the concepts that you heard about again and again today was subclinical disease and the question of outcomes that determine outcomes. And so the cohorts were rich in that. When the ADSP began, and even today, one of the new cohorts that are accruing are some of the ADSP follow-up studies that have about 40,000 people and already whole genome sequenced, and another up to 75,000 planned. But endophenotypes were not a thing. The idea was, did you have Alzheimer's or did you not have Alzheimer's? But clearly, we know that endophenotypes are very valuable. We know that they can add to power. They can give you answers in a preclinical stage because they are present in normal people as well as people with disease. And they can tell you if biomarkers are correct. They can be used for Mendelian randomization. But studying endophenotypes here brought their own problems. They brought successes like Sol1 in 2007 was associated with an endophenotype in a Framingham cohort. It took another 6 years to show up on GWAS with Alzheimer's. But then when you looked at endophenotypes of some of these cognitive functions, yes, different people had used different tests in different cohorts, and it seemed to make a difference whether you were testing memory with a list or a story, whether you were giving the stimulus visually or not. But looking at heritability, looking at the genetic basis, we were able to somewhat sort out what might be happening in the brain and what was the genetic inheritance of some of these things. You were also able to say, for instance, that genetically white matter close to the ventricle might be different from white matter further away. There was one hit on chromosome 17 that was shared between periventricular and deep, but others that were different. There seemed to be regions of white matter that led to atrophy of the brain overhead. And if you looked at the cortical atrophy and the white matter together, as one principal component, you are finding new genes. And again, you would not find these in any one cohort, but combining data in nearly 50,000 people, you are finding new pathways. And if you created deciles of the score based on these genes, it was predicting vascular dementia. This is work from Thomas Paz and many people in the Neurocharge Consortium. These endophenotypes, hippocampal volume was something that Josh led back in 2012, and this is a reiteration of that with a group called Enigma. And one of those loci is something called DPP4, and today everybody's very excited about the fact that maybe we should put GLP-1 in the water, right? And so this is being looked at in Alzheimer's as well, along with everything else. Back about just 6, 7, or 10 years ago, the cohorts had plasma. We didn't have CSF, but nobody was interested in plasma markers of Alzheimer's. It was like you have to do PET scan or you have to do CSF. But in the cohorts, we found that plasma levels of A beta 40, 42, working with Russ, who had measured it in CHS, and others who had measured it elsewhere, the same genes that determined AD risk, like ApoE, like APP, presenilin 2, were the same things that determined plasma levels of A beta, suggesting that there was a common genetic this thing. And of course, last week, the FDA approved p-tau 217 plasma assays for risk stratification for Alzheimer's disease. There's this thing called enlarged perivascular spaces. They're related to sleep. They're related to clearance of amyloid. And looking at the genetics of these, again, looked at in different ways in different cohorts, but we could put it all together because of the power of meta-analysis. We found a number of genes and merely looked at what expression would we expect to be changed with the variation we are seeing at this loci, and what are the repurposable drugs that affect the expression of these same genes. And so these may be drugs that one can look at for Alzheimer's. Also, sometimes you find completely novel variants. I showed you a locus on chromosome 17, and combining that with gene expression data and then taking it on to mouse experiments, TRIM47 on chromosome 17 seems like a druggable target, and now there are drugs being tried with colleagues in France. But Neurocharge and the principles we learned from CHS and brought to Charge have made a difference far beyond GWAS. CHS had some data on something called soluble CD14, on YKL40, and we went into a consortium called MARC-VCID. Russ was leading the vascular biomarker group, and now it's going to enter the 11th year. And by pooling data from different cohorts, we were able to show an effect with this microglial marker on risk of dementia. The cross cohorts collaboration that became C4R, that became just, non-genetic collaboration of the same cohorts that were in Charge, building on the trust and the phenotype harmonization done in Charge. And thanks to Bruce's generosity, we typically tag on the cross-cohort consortium meetings along with the Charge meetings. And now this will be about 13 cohorts across the world. And as you heard, again, in these cohorts, we're able to understand what is the thing about physical activity that is good for cognition. Maybe variations in certain metabolites. What is the thing about proteomes that can tell us about cognition? This gene called GDF15, you've heard recently about how it's responsible for cancer cachexia. It's associated with white matter hyperintensity. It's associated with executive function. So we're looking forward, the tiger has grown, and we're looking forward to exciting times. Thank you all. I want to just this was in San Antonio. Thank you.
Thanks, Sudha.